Fusion transcripts or chimeric RNAs are one thought to be generated solely by chromosomal rearrangement. Chromosomal translocations, for example, are a well-documented source of gene fusions. One of the best-known examples of fusion genes, BCR-ABL, is the product of Philadelphia chromosome. Another way to generate fusion RNAs is through chromosomal inversions. One example is EML4-ALK fusion, which is associated with a small subset of non-small cell lung cancer. Interstitial deletion is another source for producing fusions. One example is TMPRSS2-ERT fusion, which is frequently found in prostate cancer. However, recent work has demonstrated that chromosomal rearrangement is not the only mechanism to generate chimeric RNAs. Processes involving transsplicing and cis-splicing between neighboring genes can also produce chimeric RNAs. Cis-splicing between adjacent genes, abbreviated as cis-stage, is an RNA processing event that occurs within a single precursor mRNA, where the transcription machinery reads through the intergenic regions of two neighboring genes. One example is SLC45A3-ELK4 fusion. Transsplicing is another non-canonical splicing process from which chimeric RNA can be generated. Unlike cis-splicing, transsplicing joins exons of two primary RNA transcripts together. There are two types of transsplicing based on the source of primary RNA transcripts: intragenic and intergenic transsplicing. Intragenic transsplicing takes place between two copies of primary RNAs transcribed from the same genome locus. Intergenic transsplicing takes place between two precursor RNAs transcribed from different gene loci. One example is JZF1-SUG12 fusion. These findings of alternative mechanisms challenge the traditional view of chimeric RNAs are exclusively the product of chromosomal rearrangement. In addition, these non-canonical splicing products are found in normal tissues and cells, challenging another paradigm that fusion and fusion products are unique features of cancer cells. The implications of intergenic spliced fusion RNAs in cancer detection and treatment are multifaceted. On one hand, their presence in normal tissues and cells complicates the usage of fusion RNAs as a whole for cancer diagnosis and treatment. On the other hand, they represent a new repertoire for the discovery of new biomarkers and drug targets.